Hello, welcome to our channel. I am glad to welcome everyone who joined to watch the video lecture on cryoveterinary medicines. The topic of today's lecture is the effect of lower temperature on macromolecules and enzymes. What is a biological macromolecule? Well, you recall that complex multicellular organisms like humans and animals contain several different organ systems. An organ system is a group of organs. And organ is made of many different tissues, and tissues are made of many different cells. Each cell contains many organelles, and organ is made of molecules. Molecules is built from atoms, and even atoms are made of smaller bits organized together. This concept of organization is actually centers around the fact that the traits of biological structure are determined by their components. Biological macromolecules are large organic molecules made of repeating subunits. There are four types of biological macromolecules that all living things possess. These are carbohydrates, lipids, proteins, and nucleic acids. Most biological macromolecules are also considered polymers. Poly is a word part meaning many, and mer means part of unit. Polymers are specially macromolecules that are built out of repeating subunits called monomer. Mono is a word part that means one. A monomer is one unit within a polymer. When monomers join together to form a polymer, we call the process polymerization. The trait of a macromolecule depends on the trait and arrangement of the molecular subunits that it's made of. Next, let's get more familiar with the trait of the different types of macromolecules. We'll start with carbohydrates and lipids. Carbohydrates are molecules that contain carbon, hydrogen, and oxygen in a particular ratio. Polymer carbohydrates are also referred to as polysaccharide. Saccharide means sugar. We'll learn that polymers are made of repeating units called monomers. The monomers that make up polysaccharide are called monosaccharide or simple sugar. An example of a monosaccharide that you are probably familiar with is glucose, one of the substrates our cells commonly use for cellular respiration. Some of the functions of carbohydrates are storing easily transferable energy and providing structure of certain types of cells. Some complex carbohydrates include starch, cellulose, and glycogen. Let's take a moment here to note that the term carbohydrate applies both to simple sugar and to polysaccharide. But when we are talking about macromolecules, we are only referring to the complex polysaccharides, carbohydrates, and excluding simple sugars. Lipids are also of combination of carbon, hydrogen, and oxygen like carbohydrates, but they are different in many ways. First, lipids have a different ratio of this element. They tend to have more carbon and hydrogen and less oxygen than carbohydrates do. Lipids are actually characterized of their long carbon-hydrogen chain and also the fact that lipids are not soluble in water. A good example of a common lipid is your typical cooking oil, which famously does not mix with water. Some of the functions of lipids are storing energy for longer periods of time and making up the membranes of cells and organelles. Common biological lipids are triglycerides, phospholipids, and cholesterols. When we say that a biological molecule is organic, we mean that is possessing carbon-hydrogen bonds. So a molecule like glucose is what we consider organic, but a molecule like water is not. The bond between carbon and hydrogen is an especially high energy bond, meaning that the reactions that break them are usually exothermic and that excess energy can be transferred to other cellular process. Between these two types of energy storage molecule, lipids have a much higher energy to mass ratio since they possess many more of these high energy carbon hydrogen bonds. Next, let's take a closer look at proteins and nucleic acids. Proteins are extremely diverse and complex biological macromolecules. They are enzymes, antibodies, hormones, structural molecules, and much more. Proteins are polymers made of several amino acids monomers. Chains of amino acids are referred to as polypeptides because of the peptide bonds that form between them. The chains of amino acids might be folded and shaped into a functional proteins. There are 20 different varieties of amino acids, and their final sequence and arrangement that gives each individual protein its traits. Nucleic acids are DNA or RNA. DNA stands for deoxyribonucleic acids, and RNA stands for ribonucleic acids. Since the type of macromolecules is right there in the name, it makes nucleic acids particularly easy to identify. Nucleic acids are polymers made up of monomers called nucleotides. One nucleic acid can hold thousands or millions of nucleotides. Nucleic acids are able to contain a lot of information in the arrangement of the nucleotide. Also called a nucleotide sequence. They are also very stable molecules, and compared to proteins, they have a structure that's easy to replicate. This is why nucleic acids are perfect for storing genetic information. They carry huge sets of instructions for cells, and they can to copy it and pass on the offspring. Protein and nucleic acids are very closely related. You are likely already aware that DNA is transcribed into RNA and that RNA is translated into a polypeptide, which is eventually folded into a functional protein, which would make it seem like billions of years ago when life began. DNA must have existed first. But scientists interested in the origin of life know that proteins arose before nucleic acids. However, proteins are varied and complicated, so scientists theorize that DNA and RNA later developed as ways to store, copy, and transfer the information needed to make these proteins, and not the other way around. Other scientists have proposed and railroad hypothesis in which RNA existed first. We will consider proteins and nucleic acids because carbohydrate and lipid have strong covalent bond and lack special organization, contrary and secondary structure. Covalent bonds are very difficult to break, so carbohydrates and lipids are resistant to hypothermia and sublow temperatures. That is temperatures below below 0 degrees Celsius. Thus, we will consider the stability of two classes macromolecules, protein and nucleic acids. Proteins are composed of chains of amino acids. A typical protein is about 2,200 amino acids long, as there are 20 different types of naturally occurring amino acids. Many different proteins can be made. Different proteins are made of different combinations of amino acids. The sequence of amino acids in the chain determines how the chains will fold up to make the protein, so different proteins have different three-dimensional shapes. The three-dimensional shapes of protein determines its function. This is because proteins form attachments and interact with many other molecules and structures inside organisms. The shape of a protein determines that it can interact with, just like the shape of a key determines which locks it can operate. An enzyme is a biological catalyst and is almost always a protein. It speeds up the rate of specific chemical reaction in the cell. The enzyme is not destroyed during the reaction and is used over and over. A cell contains thousands of different types of enzyme molecules. It's specific to particular chemical reaction. Similar between enzymes and proteins. This is enzymes and proteins are two types of biomolecules found in the body. Both are made up of a series of amino acids. Also, there are expressed these the use of instructional of genes. Difference between enzymes and proteins. The enzymes are globular proteins, but not all proteins are globular. Unlike other proteins, enzymes can act as catalysts to catalyze and regulate biological reaction. Enzymes are functional proteins, whereas proteins can be either functional or structural. Unlike other proteins, enzymes are highly substrate-specific molecules. Enzymes are largest and most specialized class of proteins molecules. They are of two types, simple enzymes and holoenzymes or conjugated enzymes. Simple enzymes contained only proteins. On hydrolysis, they yield amino acids only. Digestive enzymes such as pepsin, trypsin, and chymotrypsins are of this nature. The holoenzymes or conjugated enzymes. This is a big group of chemical group of enzymes. They consist non-amino acids is natural along with proteinic component. They consist apoenzymes and cofactor. Holoenzymes, apoenzymes, non-protein cofactor, conjugated protein enzyme protein part plus prosthetic group. A prosthetic group is firmly bound to enzyme proteins. For example, FAD in succinate dehydrogenase is a prosthetic group that is firmly associated with the protein's counterpart. A coenzyme is a small heat-stable, dialyzable organic molecules that readily dissociate of an enzyme proteins. Thus, NAD and NADF thiamine pyrophosphatase are examples of coenzymes. Coenzymes act as donor or acceptors of groups of atoms that have been added or eliminated from the substrates. The catalytic activity of enzymes are affected by multiple factors such as temperature, water, pH, activity environment of cells, substrate, concentration of substrate, enzymes concentration, inhibitor, and product activator radiation. The most important factors in cryobiology are temperature and water. All chemical reactions take place in the aqueous medium. Therefore, if the water freezes, there will be no movement and no reaction will occur. Enzyme activity occurs in a certain range of temperature and pH. At low temperature, the number of successful collisions between the enzymes and substrate is reduced because their molecular movement decreases. The reaction is slower. The human body is maintained at 37 degrees Celsius, and this is the temperature at which the enzymes in our body work best. It is not true of the enzymes in all organizations. Higher temperature disrupt the shape of the activity site, which will reduce its activity or prevent it from working. The enzymes will have been denatured. Enzymes are also sensitive to pH. Changing the pH of its surrounding will also change the shape of the activity site on an enzyme. Many amino acids in an enzyme molecule carry a charge. Within the enzyme molecule, positively and negatively charged amino acids will attract. This contribute to the folding of the enzyme molecule, its shape and the shape of the activity sites. Changing the pH will affect the charge on the amino acid molecules. Amino acids that attracted each other may no longer be. Again, the shape of enzymes along with its activity site will change. Extremes of pH also denaturate enzymes. The charge are usually thought not always permanent. Enzymes work inside and outside cells. For instance, in the digital systems where cells pH is kept at 7 pH to 7.4 pH. Cellular enzymes will work best within the pH range. Different parts of the digestive systems produce different enzymes. These have different optimum pH. What happened to enzymes and proteins during hypothermia? Many structural and functional proteins, as well as enzymes in the composition of cells and tissues, have a certain resistance to cooling. For example, temporary cooling of the limbs or body surface does not lead to loss of functional, and hence the structure and functional of proteins are enzymes. There is a huge group of cold-blooded animals for which temperature down to 0 are not lethal. However, isolated enzymes in artificial solution are very vulnerable to low temperatures. The state of isolated protein, especially those with a quaternary structure, changes already at 4 degrees Celsius. since cooling to this temperature affects some of the physicochemical properties of water, for example, its density and viscosity, as a result of which of initial conformation of the protein's globule may change. Such transformations are accompanied by disturbance in the intermolecular interaction of the activity site of the biopolymer, which affects its function. These violations consist in a change in the rigidity of the polypeptide chains of the protein and the spatial arrangement of its individual sections. A number of oligomeric proteins experience to so-called cold inactivation upon cooling their dissociations into several subunits after cooling to 0 degrees Celsius. For example, these are enzymes such as erythrocyte glucose 6-phosphate dehydrogenase, carbamoyl polyphosphate synthetase, adenosine triphosphatase, and other. Effect of hypothermia on enzymes and metabolisms. Under conditions of hypothermia, there are no significant disturbances in the structure and organization of cells, such as during freezing. The main thing is that water remains liquid. Also, the viscosity of the cytosol may increase slightly. When the temperature of biological object decreases, from the optimum down to 0 degrees Celsius, the catalytic activity of enzyme gradually decreases, but their denaturation does not occur. The decrease in activity is explained by a decrease in the speed of movement of substrates and a decrease in the probability of their getting into the active center of enzymes. With the increase in temperature, the catalytic activity is restored again. Consequently, enzymes proteins can functional and easily tolerate temporary cooling up to water crystallization, which make it possible to maintain some cell activity during hypobiosis of organism or during storage of biological object under hypothermia conditions. A violation of the structure and organization of cells, such as during freezing. The main thing is that water remains liquid. Also, the viscosity of the cytosol may increase slightly. When the temperature of biological object decreases from the optimal down to 0 degrees Celsius, the catalytic activity of enzyme gradually decreases, but their denaturation does not occur. The decrease in activity is explained by a decrease in the speed of movement of substrates and a decrease in the possibility of their getting into the activity center of enzymes. With an increase in temperature, the catalytic activity is restored again. Consequently, enzyme proteins can functional and easily tolerate temporary cooling up to water crystallization, which make it possible to maintain some cell activity during hypobiosis of organism or during storage of biological object under hypothermia condition. Temperature is an important factor on which the rate of all enzymatic reactions depend. As the temperature rises, the rate of reaction increases. This is due to the fact that this an increase at temperature. The movement of substrate molecule accelerates and increases the likelihood of their getting into the activity center of enzymes. The temperature that provides the highest reaction rate is called optimum. For most mammalian enzymes, this temperature is a range of 30 to 400 degrees Celsius. Increasing the temperature above the optimal lead to a decrease in the rate of enzymes. This is due to changes and then lost of the normal structure of enzymes at elevated temperatures. That is, enzymes are very sensitive molecules to temperature channels. At temperature above 500 degrees Celsius, most animal enzymes undergo denaturation, the loss of their original structure. It leads to a complete loss of their activity and hence to metabolic disorders and cell functional. Thus, the rate of biochemical reaction occurring in the body depends significantly on temperature. The lower the temperature, uh, the lower the rate of metabolic reaction of cells and the organ as a whole. Accordingly, the need for oxygen and nutrient decreases, and the time that the organ can withstand in the absence of blood supply, ischemia, for example, increases significantly. It has been shown, for example, that the use of hypothermia can extend to survival times of kidney from 30-60 minutes to 13 hours. This is the basis for the hypothermic preservation and organ and tissue for use in transplantation, kidneys, heart, liver, lungs. We proceed to the freezing thawing of protein, which are more susceptible to destruction as the special structure, secondary, tertiary, and quaternary is destroyed. Let's move on the freezing thawing of the proteins. If there are more ruins, so the vast structure, secondary, tertiary, and quaternary is ruined. During freezing of isolated proteins and subsequent heating, aggregation is observed, as well as a channel in conformation, loosening of the protein globule. Freezing thawing of protein lead to dehydration of biomolecules associated with their aggregation, while maintain the secondary structure. The cryogenic stability of proteins, including membrane proteins, depend on the level of their supramolecular organization and the freezing medium in solution as part of membrane cells or tissues. Quick freezing up to minus 196 degrees Celsius in buffer solution without a significant loss of specific functional is well tolerated by simple proteins, which have stable conformation due to the ESS disulfide covalent bond of the polypeptide chains. However, due to repeated freezing and slow warming, the activity of enzymes decreases. The use of glycerol as a cryoprotectant prevents the damaging effect of freezing. The slide shows protein structures that depend on their resistance to freezing. The primary structure is sequence of amino acids connected by a strong covalent peptide bond. Secondary structure refers to the cooling or folding of polypeptide chains that gives the protein its 3D shape. There are two types of secondary structure observed in proteins. One type is the alpha helix structure. This structure resembles a cold spring and is secured by hydrogen bonding in the polypeptide chain. The second type of secondary structure in proteins is the beta-plated sheet. This structure appears to the folded or completed and is held together by hydrogen bonded between polypeptide unit of the folded chains that lie adjacent one another. The future structure refers to the comprehensive 3D structure of the polypeptidized chains of protein. Tertiary structure. There are several types of bonds and forces that fold a protein in its tertiary structure. Hydrophobic interactional, hydrogen bonding, ionic bonding, disulfide braid. And the quaternary structure refers to the structure of a protein molecules formed by interaction between multiple polypeptidized chain. Its polypeptidized chain is referred to as a subunit. Proteins with quaternary structure may consist of more than one of the same types of proteins subunit. They may also be composed of different subunits. Hemoglobin is an example of protein with a quaternary structure. Hemoglobin found in the blood is an iron-containing protein that binds oxygen molecules. It contains 4 subunits, 2 alpha subunits and 2 beta subunits. And the main factor of macromolecules during freezing. This is crystallization and recrystallization, dehydration, salt concentration, hydrogen bond breaking, violation of hydrophobic and polar interaction, aggregation. Membrane proteins are proteins situated at the lipid bilayer or membrane of the cell. Membrane protein acts as transporter, anchor, receptor, and lastly enzymes. Different types of membrane protein associated with the lipid bilayer in different ways. Transmembrane, monolayer associated, lipid linked, and protein attracted. Proteins in the membrane are stabilized by the lipid environment. When the temperature drops, lipid phase transitions occur which affect proteins. Catalytic and structural proteins built into the membrane are in a lipid environment, which is a stabilizer and regulator of their function. The activity of enzymes in membrane will largely depend both on the structural integrity of the active and allosteric center of the globule and on the physical state of the phospholipids surrounding these proteins. Lipids closely adjacent to the protein form the so-called layer of annual lipids, which contain a large number of saturated fatty acids and phase transitions of which from the liquid crystal state to the gel phase occur already at temperatures close to 0 °C. Membrane cryodamage phase transitions of lipid can lead to formation of pore. Changing in the structure of the plasma membrane would lead to damage. The ions go out from cells. As a result of phase transitions of lipids, protein can also be pushed out of membrane. Since phase transitions are lateral separation on the bulk of phospholipids occur during freezing, conditions are created for the direct effect of the solution effect on membrane proteins. Since during the phase separation of lipids, proteins can be squeezed out from the lipid bilayer. Therefore, low temperature, the main two ion transporting or receptor proteins of plasma membranes of the cytoplasmatic reticulum, enzyme complexes of mitochondrial membranes, for example, alpha-ketoglutarate dehydrogenase, increases especially strongly at temperatures of lipid phase transitions. The main two membrane-bound enzymes is especially enhancing by slowly freezing to minus 25 degrees Celsius. Since in the range of these temperatures, there are liquid microphases in the ice channels in which there are hyperconcentrated salt solution, tonicity and pH are changing. Freezing rates play an important role in ensuring the safety of proteins. Upon cooling at slow to moderate rates, crystals with a larger surface area are formed compared to crystals that occur upon freezing at high rates. When using moderate cooling rates, significant denaturation changes of biomolecules can occur. High freezing rates significantly reduce the resident time of molecules under unfavorable conditions. Therefore, their use is more preferable. When used, high cooling rate aggregation is absent or significantly reduced. This is explained both by an insufficient degree of losing and by decrease in the time spent by the system under conditional conduction to aggregation, and by a decrease in diffusion into the boundary regions. With an increase in protein concentration, its stability increases, which can be explained by a decrease in the relative amount of protein at the ice-concentrated solution surface interface. In concentrated protein solutions, even the process of denaturation of biomolecules are reversible in 85–90% of cases. Crop protections have a direct stability effect on proteins and enzymes. This is due to the fact that crop protectant allow the freezing point, prevent chorus crystallization, recrystallization, dehydration of molecules, and the effect of salts on them. Crop protection penetration into cells, for example, glycerol and dimethyl sulfoxide, are more effective since the direct effect of the macroenvironment of macromolecules. However, they can also have an adverse effect, action in high concentration on the structure and functional state of proteins and enzymes. Non-penetration crop protection, for example, starch or polyethylene oxide, preserved proteins through moderate dehydration and crystal reduction. Since after freezing solvent cells retain the ability to divide and synthesize proteins, it means that the key molecular genetic processes are preserved: replication, transcription, and translation. This in turn indicated that there are no significant changes in the structure and the properties of DNA, RNA, ribosomes, and enzymes of these processes, despite crystallization, dehydration, channels in the tonicity of the medium, and pH. Even the double helix of DNA purified from structural proteins and enzymes is not damaged by freezing and subsequent heating. Its physicochemical properties are preserved. This indicated the extreme stability of the genetic material. The primary structure in a nucleic acid molecule is very stable, as the nucleotides are linked by covalent bond. On the basis of this resistance are based the hopes of scientists to restore whose animals that have died out, but the genetic material has been preserved. For example, mammoths whose remains are decaying, hair bond. If the remains are found in permafrost, even the nuclei are preserved in the cell. According to the primary structure, it's possible to restore the genetic code, which is what the hopes for this are based on. And conclusion, protein, enzymes, nucleic acid in the composition of cells or tissues are quite resistant to hypothermia. Under condition of hypothermia, enzymes reduce their activity but do not stop working. Freezing thawing in most cases has a reversible effect on the structure and function of macromolecules. The stability of biological macromolecules makes it possible to use low temperature for cryopreservation of biological objects. Cryoprotectors indirectly contribute to better preservation of macromolecules during freezing and our lecture is over. Thank you for your attention. If you have any question, you can write me. I will be happy to answer. Thank you for your attention.